All right, so I'm going to go over a few key points with 2.1 that starts with this idea of cell theory, and we're going to go through this pretty quick. There's a lot of information. There's some calculations you can do to practice, and there are some problems in this PowerPoint. And I should note that this PowerPoint is borrowed or being used from a colleague of mine. But I didn't make this myself, but I thought this was a very detailed and um, good review of the content, and it actually builds upon what we've talked about in class. So let's go and get started. I'm going to fly through this. You're more than welcome to pause. I'm putting the PowerPoint up on Blackboard as well, so that you're welcome to download it, and I would suggest printing it and adding notes as I talk as we go through this. So just as a quick review, cell theory is three basic principles. This is the old cell theory. IB wants you to know this. So all living things are made of cells. Cells are the smallest units of life, and cells come only from other cells. So up to the first point, all living things are made of cells. That's how it's stated. And it's that multicellular organisms have specialized cells to carry out various functions. That really has to do with differentiation and cell specialization. So we have, if we use ourselves as examples, our eye cells do a different job than our heart cells that do a different job than our lung cells. So they have various functions, but they all maintain the characteristics of life. And the question that we'll talk about in just a minute is, are unicellular organisms made of cells? Well, we say that bacteria are living things, but they're not made of cells. They're single-celled or unicellular. So we'll come back to that in just a moment. The next point, cells are the smallest units of life. Organelles carry out various metabolic functions in the cell. And cell components cannot survive alone. So if we were to break apart a cell, all the macromolecules that a cell is made out of, the proteins, the lipids, the carbs, they don't survive alone. They don't meet the characteristics of life, again, which we'll talk about in just a minute. And cells come from other cells. Louis Pasteur refuted this idea of spontaneous generation with his experiments. If you don't remember what that idea is, Google it, go to Wikipedia, figure out what spontaneous generation is. But he refuted that idea, and cells multiply by division. Cells divide. And so we're going to learn about this soon. In still chapter 2, we're going to learn more about mitosis in eukaryotes. But in this video, we're going to talk about, no, sorry, not in this one, in the next one, we're going to talk about binary fission in prokaryotes. And all cells descended from simpler common ancestors. So we'll come back to this idea, but it's a little bit of the endosymbiotic theory that we've talked about in class. So just to point out, cell theory is a great example of a scientific process. Notice I didn't call it the scientific method, because there's more than one way that science progresses. But if we start with an observation, we come up with a prediction that's our hypothesis. We test that prediction. If we have data to refute it, we go back to our hypothesis to alter our prediction, change it, retest it. We collect evidence. We corroborate that evidence with lots of more evidence. We repeat it over and over. And all that evidence leads us to an explanation or a scientific theory of how all this works. So this is a long process. It started, the first person who's recorded to have been working on the cell theory is Robert Hooke. And Van Leeuwenhoek came next, and there are a series of other scientists that we're going to talk about now. So here's the evidence. So this is another statement you got to be able to tell me. You got to tell me about the evidence of the cell theory, and make sure you look at your command terms. So Robert Hooke lived 1635 to 1703, and if you remember, he's the one who coined the name cell under the microscope. Just as a reminder, we watched a TED Ed video about the wacky history of the cell theory. These names should be familiar to you. Anton van Leeuwenhoek is the father of microbiology. He discovered bacteria from the dental scrapings of his teeth. He also created the first images of sperm and bacteria. So he spent all the time looking at animal cells. He also looked at blood and pond scum. And here's a nice picture of him. This is what we think Robert Hooke looked like, but he's not a very attractive gentleman here, if I had to say so. And then for the spontaneous generation, the last idea, there's Pasteur's test. And so here are a bunch of here are a couple of videos and links. Again, I'm posting the PowerPoint. I would highly encourage you to click these, watch them, take notes, make sure you understand what's going on. But Pasteur basically prevented air from getting into his sample in one level of his independent variable. The other, it was open and airborne bacteria got in. That's our explanation, because the one that was sealed did not grow bacteria. The one that was not sealed grew bacteria. But remember Robert Remack, that from our video, he was plagiarized. His data was taken by Rudolf Virchow, who now is known for contributing to this tenet of the cell theory. All right, so here's some limitations and exceptions. Amoeba. These are single cells capable of all life processes. So this doesn't meet that first tenet that all living things are made out of cells, because it's a single cell. Same with bacteria. We're not going to really talk about the fungal hyphae because, you know, that's for HL. But muscle cells, they're multinucleate. I mean, they have several nuclei, which is interesting because they go through mitosis, make lots of nuclei, but then don't divide into separate cells. So that complicates our simple understanding. And then viruses, as you know, are debatable whether they're living or not, and whether they're cells or not. And really what it comes down to is that they can only reproduce when they have a host, because they're so small that not even a ribosome can fit inside of them. So they can't make their own proteins. So they're not using energy. Instead, they hijack the host system. So if we're the host, sometimes viruses take host of bacteria. But if, let's use ourselves as an example, if we get a virus, say HIV, HIV embeds its genetic material into your cells or into human cells and hijacks its metabolic pathway of DNA to RNA to proteins. And in essence, we are inadvertently, unknowingly making HIV proteins and making lots and lots of copies of the HIV virus, which is just a protein coating on the outside, not phospholipids like a cell membrane. And so many of them get replicated inside of a cell that it gets to be so big that it bursts open and then invades other cells and repeats the process. And then there's a link down here for a reading that essentially the idea is that if viruses can be infected by other viruses, interesting idea, does that provide evidence that they're living? That's up for you to decide, but know that IB does not consider viruses to be alive. All right, so all living things carry out the functions of life. These are the main ideas: nutrition, reproduction, movement, kind of movement, excretion, growth, respond to stimuli, and homeostasis. So ultimately, this is the idea of differentiation, that unicellular organisms that are made of one cell, like the amoeba and like bacteria, are capable of doing all of these functions. Whereas multicellular organisms have to specialize their cells so that some cells do certain jobs and so other cells do other jobs. The way I think of specialization is kind of like with doctors. You go to a certain kind of doctor for certain kinds of problems. Doctors all go through med school, and then they start to go on different tracks based on what their interest is or what specialty they want to go into. So a psychiatrist and a cardiologist they both went to med school, but they took different paths further in their medical education and training, and went in different paths and have different specialties. So that's the idea here, is that multicellular organisms have cells that do different jobs. And viruses, again, cannot carry out all of these functions independently, and so sometimes we consider them to be acellular or not made of cells. Okay? So here's another exception. Given the right conditions, cells can thrive outside of their original environment. Oops. Excuse me. Which is interesting. So this is what happens in labs. Oftentimes we study cancer in labs by growing cancer tissue, like you see in this picture. All right. This is magnification of the microscope. I'm not going to spend any time here. You can look at it if you want, but we've done a lab on this. And these are the standard SI units that you need to know, you need to be responsible for and understand. The new one that we have not talked about in class is the nanometer. but that's pretty simple. All right, and calculations in microscopy. I'm going to skip through these, but these slides are in the PowerPoint on Blackboard, and there's a series of practice questions that you can do, but there is one thing that I want to point out, this. So IB may give you just this alone and ask you what's the magnification of this scale. Well, all you need is this. So if you, let's say we look at this one up here, 2 micrometers, if the scale tells me that the distance from this line to this line is 2 micrometers magnified, if I use a ruler and actually measure the distance from this line to this line, I know the drawing size, this is the actual size, and if I divide drawing by actual, I'll find how many times it's magnified. Okay? So you can practice that if you like. So again, here's some more problems. And this is another point. Whenever you write a value, if it's less than 1, make sure that you have an integer before the decimal. So 0, would make sure you have 0. And the other idea here is that if you have 0.89, make sure you convert it into a whole number as much as possible. So in this case, this example converts it into nanometers. But if you get this far, it shows that you at least know the process. But sometimes that may not earn you the point. You have to even give it in this kind of format. So here's some more practice problems. So here's the magnification. These are just some examples. And here's some more problems. So you can actually measure the length of these cells. There are scales on each of these diagrams that you can use. And some more problems with answers. And what's interesting here is scale. So if you click on this, you actually get to zoom in to see the relative size of things. But let's look at this very quickly. Here's an animal cell that is 15 micrometers in diameter. This nucleus is 16 micrometers. The membrane thickness is 10 nanometers. The mitochondria is 3 micrometers. The ribosome is 20 nanometers. Bacterium is 1 micrometer, and the virus is 100 nanometers. So this will actually help you figure out the relative size of these components that you need to know to be able to put in a sequence. And so I highly recommend that you click on this link and explore that website. Here's some more microscopy resources. You can click on any of these links in the PowerPoint. I'm going to skip this. We're going to come back to surface area and volume ratio later. And we're going to skip this as well. So emergent properties. So this is kind of a confusing idea, but the ultimate idea is here in this blue section, that when we're looking at a whole system, we can see that an organism is more than the sum of its parts. In biology, historically, what we used to do is called reductionist biology. Basically, we take everything apart and study it individually. Kind of like when we watched the video, the documentary on Forks Over Knives. They talked about these mechanisms of how these proteins and carbs and lipids work. But really, the idea is how all of this works together. I think the analogy in the movie that Dr. Caldwell used was it's a symphony. All of these things working together. So emergent properties are seen at every level of increasing complexity. So every time we add components, we see that there are emergent properties, or things that are working beyond the simple individual components. So when we move from atoms to molecules, we see emergent properties. A molecule is not just this, doesn't carry the same properties that each of the individual atoms in that molecule has, even to the cell, the organism, and the biosphere. And one thing affects the other. So as we move from cell organelles to cells to tissues to organs, organ systems to organisms, we see increased levels of complexity in their function. And one problem in a lower level of complexity can have a detrimental effect on the larger system. Those are the emergent properties. That's all it is. And so here, this is what's interesting about evolution. We haven't talked about evolution in detail yet, but there's this book called The Blind Watchmaker, and you might have heard this argument before against evolution. You might have heard this argument that living things are so perfect, how can they work so well without a watchmaker? Well, the analogy here is that over millions of years with infinite mutations and combinations, and it is inevitable to have complex structures. And as we add more and more complexity, those properties become more and more emergent. They become more and more, for lack of a better word, amazing. And so there's no purpose or design to evolution. Instead, they're beneficial mutations in a particular environment that will allow the organism to survive and reproduce, and therefore pass down that genetic material that will code for that same mechanisms, those same mechanisms and emergent properties. All right, so last topic, cells. These are keywords that you need to know: totipotent, pluripotent, multipotent, unipotent, and nullipotent. So we all start from one single cell, from conception, from fertilization, and that single cell is totipotent. It can become any cell type. And the next few cells are totipotent. But then they become pluripotent. And then they can only become certain kinds of cells. They've narrowed their choices, I guess, if we consider them to be conscious beings. They've narrowed the choices of things that they can become. Multipotent is even more narrow. Unipotent can only become one cell type, and nullipotent can't divide anymore. So we have certain cells that are multipotent and unipotent in our body right now. So when you get cut, your skin gets cut, your body repairs itself, and those cells can be considered multipotent, or probably more so unipotent, because they can replace the skin cells. Nullipotent are red blood cells because they don't have their own DNA. They had DNA. They made so much hemoglobin or protein that they've kicked out all the other organelles in their cell body, and now they're just protein, so they can't replicate on their own. Instead, they need bone marrow from your bones. And the idea that cells can become different kinds of cells is differentiation or cell specialization that I've alluded to before. So differentiation depends on the activation of genes in sequence, often triggered by environmental changes. So if we change the environment, we can change what kind of cells these stem cells become, which is an interesting idea. But as you know, embryonic stem cell research is controversial in our larger society because of what that means coming from an embryo. But there's lots of research done on adult stem cells. But remember, adult stem cells are going to fall into this category, whereas embryonic stem cells fall into this category, which we can't really, of totipotent and pluripotent, which, again, funds are limited for that kind of research. So cell differentiation is the expression of different genes. So here's another big idea. All cells in your body carry the same genes in their nuclei, except for your sperm and your egg. So what makes a cell different is which genes are expressed or turned on and off, which also, if you think back to a previous unit, means which genes are coding for mRNA and therefore protein, and which are not. So I kind of think of it as each cell in your body is carrying the entire series or volume of the Encyclopedia Britannica, but each kind of cell is only reading one book for instructions. And this is triggered by changes in the environment around the cell. Sometimes it's even the order in which the cells are aligned and which cells are their neighbors, which is kind of an interesting idea. And there's stem cell transplants. So you need to be able to know some of them, and one of them is a bone marrow transplant. You might have heard of bone marrow registries, or really this is for leukemia or bone marrow disease, and sometimes stem cells can be harvested from bone marrow of someone who doesn't have that cancer and replaced into that of someone who needs healthy bone marrow. And those those produce healthy red blood cells for the patient. But remember, bone marrow would be considered a type of stem cell because they can make code for either more bone marrow or red blood cells. And then we can clone. We can clone in labs based on that. So here's some links. And here's some more information, but I'm going to go through it and let you read on your own. Some more resources and some more resources, and I think that's it. Yep.